Hi, my name is Raquel Fleskes. I am a postdoc in the Department of Anthropology at the University of Connecticut. I'm really excited to be here today to record and present to you my project pitch for the UConn Postdoc Seed Award. My pilot project is on the extraction of DNA from clay tobacco pipe stems, and I actually have a replica of one of these clay tobacco pipe stems right here. These clay tobacco pipe stems are a common artifact in the colonial archaeological record, and they were used by persons of European, African, and indigenous descent to smoke tobacco or other plant materials from. Now, the reason why these guys are really common in the archaeological record is because of this clay content or this clay material. The clay preserves really nicely in the archaeological record. And in addition, this clay also preserves DNA really nicely. So the clay silica structure has been shown to protect DNA molecules through time and basically preserves those nucleic acid, because the DNA likes to bind to the silica particles present in the clay. About 3 years ago, a paper was published on the extraction of DNA from these clay tobacco pipe stems. It was done by a historical archaeologist as well as folks from the University of Illinois and the University of Copenhagen, and it really was a transformative paper. However, only one of 3 of their samples that they tested was successful. And when I took a closer look upon that paper when it came out about in 2019, I found that they used an extraction and purification protocol that was really meant for modern DNA or DNA from living individuals. Now, the DNA, what we call ancient DNA or DNA from archaeological artifacts, which has been my field of study, what I did my PhD on, is usually typically highly degraded. So it's usually found in really small fragment sizes, and there's usually not a lot of it. And also it can have things like inhibitors or things that block our DNA from getting extracted, amplified, and sequenced. So when I originally came across this paper, I thought, well, you know what if we could extract this DNA from these clay tobacco pipe stems with more efficiency by applying methods of DNA extraction typically applied in ancient DNA contexts. So, for instance, in my PhD research, which focuses on doing ancient DNA in colonial period North America, my work uses ancient DNA extraction protocols for human skeletal remains. So the plan for this pilot project is to compare different methods of DNA extraction to determine the most efficient way to obtain and extract DNA preserved in these clay tobacco pipe stems. The reason why this is so important is because the DNA trapped in these pipe stems can provide information on the users of these pipe stems. So the DNA that we can extract from them can be used to generate genomic profiles of its users. These genomic profiles can inform things like ancestry or where you come from, relatedness, biological kinship. If we see relatedness between different of the users that are using different pipe stems, this might help us think about kinship in site occupation, as well as it can inform things like phenotype or physical expression of our genes. So this can inform things like skin color or health status. So this information is really important for us to deepen site interpretation in archaeological contexts, and has a lot of impacts for how we think about the past, how we think about human history. So for instance, with this study specifically, it's going to have a lot of local impact for our understanding of local Connecticut history. The 8 pipe stems that are going to be used in this project come from sites local to Connecticut, spanning the 16th to the 18th centuries. One of these is in Glastonbury, about 30 minutes away, and the other ones are from the coast. And so doing this study, we could potentially extract the DNA of users of these pipe stems to understand things like how many people were occupying these sites. Are they European? Are they African? Or are they indigenous? And this will allow us to understand the histories of these sites in a way we've never been able to do before. In addition, this project is really important because it can open up a new and novel avenue for efficient extraction of DNA from ceramic objects. These ceramic objects are found across the globe. A lot of ceramic objects are made out of clay. And so if this project is successful, which I think it will be, we can open up a new way of extracting DNA of these past archaeological populations. So I would like to thank you for watching this video. I really hope this project gets funded. I have been waiting for an opportunity to really pursue the methodological development of DNA from archaeological artifacts since I read that paper in 2019. And I really think that this could be a hugely transformative method for anybody in anthropological genetics, archaeology, or any sort of way of thinking about how we study the past. So thank you very much.